Good afternoon, everyone, and welcome to today's webinar on rapid analytical approaches for biologics characterization and mass photometry. My name is Declan, and on behalf of Oxford Global and Riffin, I would like to thank you all for joining us today. After the presentation is complete, we will have a 10-minute Q&A session, so please submit your questions through the question panel on your menu display throughout the presentation. The full recording of this webinar will be made available to download on our secured website, and the details will be sent to you shortly after today's session. Today's presentation will be given by Dr. Rasha Mahed, who is a technical sales specialist at Riffin, and today's webinar will cover mass photometry and provide insights into the capabilities of the technology for characterization of therapeutic antibody samples. Rasha Mahed is a sales application specialist at Riffin, a company specializing in the development and manufacturing of instrumentations for the characterization of biomolecules using mass photometry. She has over 5 years of experience in the biotechnology industry as a microbiology and evolutionary biology researcher, and Rasha has a PhD from the INRA French National Institute before joining Riffin. Rasha also completed a postdoc at the University of Cambridge, designing genetic modification strategies for viruses and pioneering phage display techniques. So thank you, and over to you, Rasha.
Thank you for the lovely introduction. Good morning, good afternoon, good evening, depending on where you are based in the world. Thank you for being here and for your time today. So today I will be talking about mass photometry, which is a rapid analytical approach to characterize your biological samples. Before jumping in and talking about mass photometry, I would like to say a few words about us, Refine. So Refine, it's like at Refine, we pioneer in mass photometry. We are the first and the best to provide mass photometry for the scientific community. We have been in the market for a few years now, more than, I would say, 5 years, but we have been successfully adopted by industrial and academic partners all over the world. We have now more than 350 systems installed worldwide. Our headquarters are in Oxford, UK, as you can see here in this small dot. However, we have presence worldwide in the world, in America, North America, in Boston, and as well in APAC, in Japan, and in Singapore. So mass photometry, it's an innovative, novel biophysical analysis tool that enables you to be applied in numerous or various school research projects. So it is a technology that will provide you a rapid and accurate mass measurement of biomolecules in solution in their native states. So your molecules will be in their native behavior. You don't need to label them or extra treatment. It is a single molecule approach. It means that you have a single molecule sensitivity, so it's not a bulk measurement, and that will allows you to access subpopulations to determine complex stoichiometries, or if you have a protein that has different oligomeric states, you can check mass photometry to determine the abundance of each oligomeric state in a sample. It allows you as well to monitor complex and multi-step processes. And as well, as I mentioned before, it's a single molecule approach, so it's not a bulk measurement. So it allows you to detect low abundance species or rare species in your sample. And it can characterize and monitor sample homogeneity or heterogeneity. How mass photometry works. So this is a very high-level overview on how the technology works, of course, because the physics behind this is much more complicated than that, but this is just an overview about how the system works. So your experimental setup, it's a glass surface. It's a cover slip that Refine provides. So you have here, as I mentioned, your glass surface, and then you would come and you pipette a drop of your sample on top of this glass surface. So your sample or your molecules are in solution. And the solution is your buffer of choice. So we are flexible to variety of buffer. So you have here your molecules in solution, and then what happens afterwards is that we shine a laser to illuminate the sample. So we shine a laser throughout the sample. Part of the laser will be reflected by the glass surface and then collected by the objective. And since the molecules, they are in solution, they diffuse around to come close to the glass surface. And when they actually bind, and specifically to the glass surface, is where they will scatter light. What we quantify is the interference between the scattered light and the reflected light, resulting in a contrast that you see here. So that contrast scales linearly with mass, meaning that if you have like a molecule of higher molecular mass, you would expect a stronger contrast than a molecule of lower molecular mass with a beamer contrast, as you can see here. It's a live image. It is a recording or a movie or a film that we record for a minute of time, 60 seconds. This is how long it takes to do one acquisition with mass photometry is 60 seconds, sorry. So then you take this acquired data to our analysis software, and then as an output, you would have a histogram like so, a mass histogram, because the software will go throughout the acquired data. It will count each contrast presented in the acquired data, because each contrast, it's resulting from one molecule in the solution. It will assess the ratiometric contrast, or the contrast that we observe here, and it will be plotted like so. So here you observe on the x-axis molecular mass, and then on the y-axis, number of counts. So number of counts, number of events, number of biomolecules, they all mean the same. One count is one biomolecule or one particle in your solution. And then you have here your distribution, your mass distribution. We have here 3 different mass distributions because we have started initially with a sample with 3 subpopulations. By fitting Gaussian fits to those mass distribution, you'll be able to get the mass, molecular mass of each peak alongside the relative abundancy or the relative concentration of each peak within a sample. What we quantify with mass photometry, it's molecular mass. However, we have a wide dynamic mass range. So we can quantify everything that goes between 30 kilodalton, so this is our lower mass limit, going up to 5 megadalton. However, that 5 megadalton, it's not a hard cutoff. So if you have something above 5 megadalton, you will still able to see with our technology. However, between 30 kilodalton to 5 megadalton is where we guarantee our precision in mass. So above higher megadalton, you expect higher error on the mass attributed. So in this mass range, it's a very dynamic mass range. Everything basically that fits into the mass range, you can quantify it with mass photometry. That means you can characterize your protein sample, your antibodies, your protein complexes. You can as well determine and characterize your nucleic acid fragments. It could be DNA or RNA, as well as small viruses such as AAV, all within our capabilities. This is a sneak peek to show you how the system works, just as well to mention and to demonstrate how easy it is to use it and how it is very, very user-friendly. And as well, it shows how the system as well has a very small footprint, and it could be installed in any lab, in any labs. So I'm gonna just play a quick video. So you open the lid and you can see here, this is the glass surface that I have mentioned. You pipette a very low amount of samples. You need few microliters from nanomolar range on this glass surface. As you can see here, we see this droplet of your sample pipetted on this glass surface. You close the lid, and then automatically when you close the lid, you would see an image like so on our acquisition software. Each dark spot or point that you see here is the contrast that I have mentioned before, and the intensity of that darkness or the blackness or intensity of that contrast is what we quantified, and that would scales linearly with mass. So after pipetting, a user would come and will press on record, and that with that, the system will record 60 seconds of data, and that's basically how one mass photometry measurement looks like. As I mentioned, it's a very, very straightforward procedure, and it requires very low amount of like expertise as well when measuring with mass photometry. So we give a molecular mass. So if you think about it, anything that's gonna change the mass distribution within your sample, we were able to quantify it with mass photometry. So mass photometry helps you to answer most of the biophysical characterization questions. So for example, it could be a simple sample characterization. You want to check on the quality of your sample, to assess if it's pure enough for your next analysis, or you can as well, if you're working in purification processes or productions of proteins or biomolecules or biologic in general, you can as well optimize the condition of your protein production or your biologics purification. You can as well do interaction studies. You can assess how strong are your interaction, what is the complex stoichiometries, and as well you can analyze your complexes. So mass photometry as well opened the door to many cool biophysical characterization questions, and as well can help you have a more streamlined biophysical characterization of your biologics. I will start first with purification. So this is for a 20S proteasome purification process, but could be applicable to any type of biologics. This type of a complex proteasome follows 5 purification steps, and you can see here, and the peak, corresponding to the proteasome, it's around 700 kilodaltons. So what we are seeing here in this different plots, so this is like the process, the purification process, is the enrichment of the proteasome throughout the process. So you can see here in this highlighted area where the proteasome is, and you see how it is being enriched throughout the purification processes. However, as well, we can give the relative abundancy of the species compared to each other. If you look at the molar fraction of the proteasome compared to the contaminants, as you can see here, we observe a decrease in the, quantity or the molar fraction of the proteasome from step number 4 to step number 5, meaning that Wiz Institute perhaps should be stopping their purification step at step number 4, where they have the most of their biologics or their biologics of interest in the solution. As you might know, when you're developing and producing therapeutics antibodies, people face antibodies aggregation issue. This is the main issue observed in this particular field. This is a challenge. It can affect the efficacy and the quality and the efficacy and the quality of the therapeutics. So basically, mostly fragmentation and aggregations are the key quality attributes when we are talking about biotherapeutics that like antibodies producer or manufacturers need to monitor as well, because they need to monitor the quality of their products or their therapeutics. So in the next couple of slides, I'll be discussing how mass photometry can help you streamline those processes and efficiently characterize aggregation and fragmentation of those antibodies. So in this data set I'm going to be presenting, our industry collaborator wanted to understand the effect of different stress factors, which are light, pH, and peroxide, on a panel of 14 antibodies, and looking particularly for those quality attributes, which is aggregation and fragmentation. I just as well wanted to mention that each, experimental setup or each condition for those 14 antibody was repeated in triplicate. Therefore, in total, they have around 168 measurements. These type of experiments would be perfect for the automated version of mass photometry. It's a product that I will be discussing later on during my presentation, because if you are running such a volume of experiments, it would not be ideal to manually or to have someone to manually pipette 168 sample. So that's why later on I will be explaining how we can gather those type of experiment with our automated version of mass photometry. So for the sake of time, or else we'll be here all evening and you guys are gonna hate me for it, I'll be just presenting the result of one antibody of the 14. So this is, as I mentioned before, it was done with our industrial collaborators. So we weren't told which stress condition correspond to, which stress condition correspond to light, pH, and peroxide. So they are labeled as stress 1, stress 2, stress 2, and stress 3. However, you can look at the histogram and you can compare the level, you can compare the effect of the stress like on the 3 different stress compared to the control. So I will explain quickly how the result looks like. So if you can see here on the x-axis, we have molecular mass, and on the y-axis, we have number of counts, so number of particles, and number of antibodies. The area, the histogram highlighted in blue, it's the monomer or the intact antibody, and then the area highlighted in orange corresponds to the lower mass molecular or the lower mass species, which could be the fragmentation of the antibody. And the areas here highlighted in gray correspond to the higher order species that could be the aggregation. So here we have a trimer and a dimer and a trimer. So what you can see here is we have the control, we have very low percentage of aggregates, and then you will have a different stress factor in here. What you can do, you can export, yes, it's correct, the word I'm looking for, export the number of counts of each of those species. For example, the lower mass and the higher mass correspond to the aggregate, and as well the percentage of those subpopulation within this type of sample, and those values are plotted here in this table. So you have here the control, it has around 95% of intact antibody, and you have the percentage of the fragmentation and the aggregation, and you can see the different stress level, how much aggregation occurred on those different stress levels. So if you're looking at the aggregation or the higher order molecular species, you can see that stress number 1 and stress number 3 gave the highest or the largest fraction of dimers. However, if you want to look, let's say, on the fragmentation, you can see that the stress number 2 resulted on the most fragmentation of your antibodies. Continuing on this note, SecUV, it's used in the field for sample characterization and quality attributes. So this is how mass photometry compares to SecUV. This work was done with RIC Biologics in Belgium, where they have tested 5 antibodies. So you have the trastuzumab originator and 4 biosimilars from 4 CHO clones. The supernated from the CHO clones were purified using protein A affinity chromatography and then analyzed by SEC, where you see here you have the chromatograms coming from the SEC, where you can see the monomer peak here. Okay, so I forgot to explain the plot. So on the x-axis, you see the elution time, because on SEC, the larger species will elute before let's say the larger species will elute first, followed by the smaller species. And here you see the UV absorbance on SEC. So what you see here, it is the main peak is the monomer. However, if you zoom in on the left side of the plot, you can see the different the dimers of the different populations, like the 5 different antibodies. And here you see perhaps fragmentations, because you have a lower mass species. Here you have maybe fragmentation or contaminants in the preparation. So if we look at the dimer here, you have almost none for the originator, the herceptin, and you look here, you have different level of dimerization depending on which clone it is. So I would say clone number 10, well, by just observing here, no, clone number 10 and clone number 3, I would say gave the highest number of dimerization. So how this compares to mass photometry? So we have run the same sample where as well, again, measured with the mass photometry. So what you see here in this on the right side of the plot is the mass photometry data, where on the x-axis you have molecular mass, and then on the y-axis you have number of counts. On the first plot here, it is the originator, the Herceptin originator, and here you have the four different clones. In blue, you have the intact monomer, and in orange here you have the dimers, the dimer of the different antibodies. But however, I want to highlight one point here. For quantitative comparison, we have to all first agree that we cannot compare UV absorbance, which is what UV gives, compared to single molecule counting, which is what mass photometry gives you, which is number of counts. Therefore, to interpret the result correctly, the SEC-UV data needs to be corrected or normalized. So the UV absorbance value generated here using molar extinction coefficient in accordance with the Beer-Lammer law in order to get relative abundances of these species. And then those relative abundances could be then correlated or compared to mass photometry. If you need more information about that, we have an application note, or sorry, a tech note dedicated for this particular data, where you can go and download it from our website as well. So after doing this type of normalization, those data are plotted here on those two tables. So you see here the data obtained from SEC-UV, and you see here the data obtained from mass photometry. You see the originator, the trastuzumab originator, the monomer on the dimer, the percentage, and different clones from different clones from CHO cells. So here you see the dimer coming from each population. So you see in clone 2 and clone 10 have the highest fraction of this dimer, and that correlates very, very well with mass photometry. So as you can see here, mass photometry and SEC-UV data correlate very well. So mass or monoclonal antibodies aggregation can be quantified using both technologies, SEC-UV and mass photometry. And as you can see here from this set of data, the data correlates really, really, really well. However, I just wanted to mention that for one SEC-UV measurement, 10 microgram of antibody is required. However, for one mass photometry measurement, you would need only 30 nanogram of your sample to run one measurement. For mass photometry measurement, as I mentioned before, it's only 1 minute of time without any pre-treatment or any pre-preparation of your sample. However, it's a minimum 30 minutes for one SEC-UV measurement. So as well here with mass photometry, although you get comparable data to SEC-UV, you are getting the information in much more shorter time and using much more less sample. And as well, mass photometry is not a column-based technology, so you'll get your result in a more native approach. So how we talked before about sample characterization and quantifying quality attribute, but how as well, mass photometry can also be used to characterize antibody-antigen interaction. So what you see on the top plot here is Herceptin, the antibody Herceptin and its receptor HER2 measured separately. So you can see here the peak corresponding to the Herceptin, and then the antibody, its receptor, or the antigen, which is HER2. And then what happened is that we mixed those two components or compounds in different molar ratios, so together to promote the interactions. So in blue, you see the one-to-one ratios, where you see that you have one-to-one stoichiometries as well, which is one binding site of the antibody occupied by the antigen. And then when you mix the proportion in 1:2, which is one antibody to two antigen, you are shifting the equilibrium to this state where you have the two binding site of the antibody bound or occupied by the antigen. So you get as well an idea about the stoichiometry of your interaction. As I mentioned as well previously in my presentation, mass photometry requires a very low amount of sample, which is nanomolar range, which is maybe not suitable for low affinity interactions. However, recently we have launched a new model, Mass-Physics HC, which allows a very rapid dilution, which is going to be the ideal system to study low affinity interaction. This is a new model. It's an add-on to MP, so it's not a new system. It's just an add-on, consumable add-on to any existing mass photometer. So here we give an example about IgG and its receptor, the Fc receptor as well. So if you look in if you look in orange, it's a normal mass photometry measurement where we barely see any interaction. So you see here the antigen. This peak corresponds to the antigen, and you see here the antibody. However, where we are expecting to see interaction, we don't see anything when you do manual dilution with the normal manual mass photometer. However, when you do it via the Mass-Physics HE system, which is the rapid dilution, you observe completely different results as well, because those type of interaction requires higher concentration. So you see here, you still see the monomer of the antigen, as you can see here. However, we see in the rapid dilution the dimer of the Fc, which we lost it when we run a manual system. We as well quantify the antibody, the monomer, and we start to see interactions of those two components. So you have 1-to-1 here. So one antibody to one antigen, and you have here 1-to-2 stoichiometries, which is one antibody to two antigens. So with the new add-on, which is mass fluidics, we are able to extend the mass photometry concentration and as well widen up our scopes to as well be able to quantify low affinity interaction, which was not possible with our previous setup. With mass photometry, as well, you can quantify interaction and dissociation constant by doing a single shot KD measurement. Here, in this case, we are looking at a glycosylated IgG and its Fc receptor. So, like the two components will run separately and then will run mixed together. So here, here would happen that each component, again, as I mentioned, is measured separately and then together. And by quantifying the free antigen here in this area highlighted in gray, the free antibody in this area highlighted in as well gray, and then the compound to the complex or the interaction in here, you can extrapolate or export those number of counts, convert them to a concentration, and then you're able to estimate your KD. So you can, as you can see here, this KD, so we have a very high affinity interaction because the KD is very 0.05 nanomolar. So similarly, we have done the same experiment, however, with a deglycosylated IgG. And when we done the same, so the plots can read the same, we observe a different type of behaviors because we observe a decrease of 20-fold in the binding affinity. So the binding affinity is 24 less from the antibody towards its antigen. So you can as well see the effect of glycosylation on the binding affinities of those two components together. So just to give a quickly overview that as well with mass photometry, we can able to characterize viral vectors such as AAV and larger viral vector as well. So for the people who are interested in learning more about how we can characterize AAVs with mass photometry, please do reach out to us and we are happy to expand on that particular topic. However, I just want to give you how we read the result or how we can characterize AAVs with our technology mass photometry. So our technology is a single molecule approach, meaning that each AAV in your sample will be quantified by MP and mass photometry, and since it is a single molecule, since it is a technology based on molecular mass, we are and not size or hydrodynamic radius. We are the perfect tool to distinguish between empty, partial, and full, because the difference between those population is purely molecular mass and the mass of the cargo that you are packing inside of your capsid. So here, just to quickly show you that with mass photometry, you can consistently measure throughout the whole range of full AAVs, going from 0% AAVs to 100% AAVs. Again, if you want to learn more, just feel free to reach out to us. But again, the same principle applies from biologics. You require very low amount of sample and a very low titer or concentration, which is ideal in an AAV concept because they tend to be very precious sample that we don't want to waste a lot of that sample on analytics or analytical characterization. So to recap, mass photometry, it is a single molecule approach to characterize biomolecules or biologics in solution. They are in their native state without any need of labeling. It is a very rapid characterization, so we need 1 minute to acquire data and few minutes to analyze, so you have a full understanding on your sample within less than 5 minutes. It's a solution-based technology, and as well, we have a very wide dynamic mass range that allows you to study different oligomer state, different complex composition. You can as well track multi-step processes, or you can do as well equilibrium studies. So we have been here, well, not here, we have been in the market for 5 years. So we have now few system on the market. So please meet the family of mass photometry. The 2MP, it is basically what I have presented throughout my presentation. It is the second generation of mass photometer for single molecular mass measurements. We have the automation version of the 2MP, which is a product that I'm gonna discuss later on, or elaborate later on in my presentation. And we have the SAMX MP, which is a product that it is dedicated for AAV characterization. So if you're interested in that particular application, please as well reach out to us for a more in-depth discussion. The 2MP has a mass range from 30 kilodalton to 5 megadalton. We require a very low amount of sample from let's say we quantify from 100 picomolar to 100 nanomolar. This upper limit could be extended to 10 micromolar with the mass physics add-on. We have as well a very good resolution on our system, and our mass error, it's maximum of 5%. So when I mentioned about the 168 measurement early on in my presentation, that those type of experiments are ideal for automated mass photometry, this is the product that I was talking about. So the same advantage, all the advantage that you get from a manual mass photometer that I have presented today in my presentation, you can add on top of that automation. So in this case, you have a greater data confidence, reduced operational time, and as well allows you to have a more robust, let's say, type of measurement because you are removing the human error from your measurements. So the automated mass photometry allows you to have a rapid type of measurement, operator free, where you can run 24 measurements without any user interference throughout for 90 minutes of time. So that is the automated mass photometer. Again, as I mentioned before, this is how it looks like. So you have the same MP setup, so it doesn't take much more space or bench space on your bench. However, you have it's like it takes more in height rather than in width. So in this case, this is how it looks like from the inside. So you have here still your objective that you saw before. The samples are pipetted out by a liquid handler from a 96 well plate. Here you have your tips, and here you have a bin to eject your tips. So again, it is an automated approach to mass photometry. It is still a label-free measurement. You can do 24 samples in 90 minutes. And here, by removing the human error, you can have an increase in data reproducibility. And as well, it's much easier to use because you just need to set up software and leave and come back with your data already acquired and analyze at the same time. Thank you so much for your attention. Thank you as well for being here and for your time attributed. If you have any question, I'm happy to take that on, and we can have as well a more dedicated discussion based on your particular questions and projects. Thank you.
So thank you very much for your time today. And all that remains to be said is have a great day. And we look forward to working to one of our future events. And thank you again, Resha, for your presentation and particular day.
Bye-bye.
Thank you, everyone. Take care. Bye.